in systems biology, that is cluster analysis for microarray data. So in this study, I will be mainly focusing on cluster and how to work on clustering when you have microdata. So the aim of clustering here is to group objects according to their similarity. A set of objects that are similar to each other and separated from the other objects are called clusters. So this is raw data, and then green and red points were generated from two different normal distributions, which you can see clearly. So red groups are cluster 1, then green are cluster 2, depending on. So you can also make more clusters by assigning the threshold or cutoff for this y-axis values. Now, clustering microarray data, genes and experimental samples are given as the row and column vectors of a gene expression data matrix. Clustering may be applied either to genes or experiments. So, why cluster? We cluster the genes, identify groups of possible co-regulated genes, for example, in conjunction with sequence data. Identify typical temporal or spatial gene expression patterns, that is cell cycle data. Arrange a set of genes in a linear order that is at least not totally meaningless. Now, why cluster? We cluster the experiments or experimental samples. Quality control, detect experimental artifacts or bad hybridizations. Check whether samples are grouped according to the known categories. Then identify new class of biological samples, for example, tumor samples or subtypes. Now, here you can see the different groups or clusters are present. For example, lymph node proliferation, activated T cells, and then germinal B cells, then pan B cells. So you can see these are the different clusters, different subclusters. Here you can also see the clusters and subclusters. For these things, you just need to work on the threshold of these distances or the parameters which you have used for clustering. Now, generally, cluster analysis is based on two ingredients, that is distance measure or the algorithms. So quantification of dissimilarity of the objects, that is distance measure, or similarity of the objects. Now, clustering algorithm, a procedure to group objects, aim is small within cluster distances, large between cluster distances. Now, there are several algorithms which I have discussed in data science tutorial, and you can also have a look about the details of the algorithms and different types of clusterings. Now, some of the distance measures are Euclidean distances, Manhattan distances, correlation distances. Depending on your data, you can choose the types of distance which you want to calculate. Now, which distance measure to use? The choice of distance measure should be based on the application area and what sort of similarities would you like to detect. Correlation distance, DC, measures the trends or relative differences. And next is which distance you want to use. For example, Euclidean and Manhattan distance both measure absolute difference between vectors. Manhattan distance is more robust against outliers. Many people may apply the standardization to the observations, subtract the mean and divide by standard deviation. After standardization, Euclidean and correlation distances are equivalent. Now it comes what is k-means clustering. This is the also thing which I have explained in details. So k-means clustering, here the input is n objects given as data points. Then specify the number of clusters. Then initialize the number of clusters and then centers iterate until convergence reach. Assign each object to the cluster with closest center, and then the centroids or mean vectors of the obtained clusters are taken as new cluster centers. K-means clusters can be seen as an optimization problem, and for which you need to go for minimization of the sum of squared within the cluster distances. Results depend on the initialization. Use several starting points and choose the best solution. Now, what is partitioning around midwives or PAM? K means clustering is based on Euclidean distance. Partitioning around PAM generalizes the idea and can be used with any distance measure. The cluster centers or prototypes are required to be observed to the observations. Try to minimize the sum of distances of the object to their cluster centers by using an iterative process that analogous to one of the K-means clustering. So, how to choose the number of clusters? There is no straightforward rule. It depends on what you want and when you feel that the solution has reached. Many heuristic approaches try to compare the quality of clustering results for different values of clusters. So, the problem can be better addressed in model based clustering, where each cluster represents a probability distribution and a likelihood based framework can be used. Now, it comes to SOP or self organizing maps. So here, K equals R into S, that is, clusters are arranged as nodes of two-dimensional grids. Nodes represent cluster centers and or prototype vectors. This will allow to represent similarity between clusters, and the algorithm initialize nodes at random positions and iterate randomly, pick one data point, move toward nodes towards X. The closest node, most remote nodes are less. Decrease the amount of movements with number of iterations, and here you can see the references. Now you can go for the detailed about it with the real application for experimental data in the paper by Tamayo et al. 1999. Now, hierarchical clustering, similarity of objects is represented in a tree structure that is dendrograms. And Golub's data, which was published in Nature, you can also have a look over it, which clearly shows the implementation of hierarchical clustering. And here, different types of leukemias were presented or studied, clustering based on the 150 genes with highest variance across all the samples. The advantage: no need to specify the number of clusters in advance, nested clusters can be represented. Now it comes to agglomerative hierarchical clustering, bottom up algorithm, and here you can think about start with objects as clusters. In each iteration, merge the two clusters with minimal distance from each other until you are left with a single cluster comprising all the objects. But what is the distance between the two clusters? This is what you need to understand. So calculation of the distance between two clusters is based on the pairwise distance between members of the clusters. Complete linkage, largest distance. Now, average linkage is average distance and single linkage is smallest distance. So you need to think about how complete linkage gives preference to compact or spherical clusters. Now, hierarchical clustering here, the height of a node in the dendrogram represents the distance of two children clusters. Loss of information. N objects have this combination. Pairwise distance. Tree has n minus 1 inner nodes. The ordering of the leaves is not uniquely defined by the dendrogram. So, this you can also understand in the same paper which was published by Golub et al. In Nature. Now, alternative or direct visualization of the similarity distances. Here you can also useful if one wants to investigate a specific factors. For example, advantage is here no loss of information. Sort experiments according to that factor. The role of feature selection. Some people first select genes that appear to be differentially expressed between the groups of samples. Then they cluster the samples based on the expression levels of these genes. Is it remarkable if the samples then cluster into two groups? Now, this does not prove anything. Because the genes were selected with respect to the two groups, such efforts can even be obtained with a matrix of IID random numbers. Now, clustering of time course data. Suppose we have expression data from different time points and want to identify typical temporal expression patterns or profiles by clustering the genes. Usually, clustering methods or distance measures don't take that ordering of time points into account. The result would be the same if no the time points were permuted. Simple modification. Consider the difference between consecutive time points as an additional observation. Then apply a clustering algorithm such as K-means clustering to the augmented data matrix. Now, by clustering, usual clustering algorithms are based on global similarities of rows or columns of an expression data matrix. But the similarity of the expression profiles of a group of genes may be restricted to certain experimental conditions. Goal of biclustering is to identify homogeneous submatrices. Difficulties, computational complexities, assessing the statistical significance of results. This you can also see in detail in the paper by Tane et al. 2002. Now, in conclusion, clustering has been very popular in the analysis of microarray data. However, many typical questions in microstudies, for example, identification of expression signatures of tumor types are better addressed by other methods, for example, classification, statistical tests. Clustering algorithms are easy to apply and they are useful for exploratory analysis, but it is difficult to assess the validity or significance of results. Even random data with no structure can build clusters or exhibit interesting looking patterns. So, in R, if you are working, there are a number of libraries which you can have a look. And similarly, in Python, Perl, and MATLAB, many inbuilt and reliable functions are available for different types of clustering. So now you can also look for the